Hello everyone, welcome to Biocool classes. Today's topic is protein structure. So we will be learning about protein structure in this class. Okay. So biomolecules, and so first, before going into protein structure, let's try to understand the concept of biomacromolecules. What are biomacromolecules? So biomolecules are, what are biomolecules? Biomolecules are all kind of compounds which have carbon. So a biomacromolecule are very big biomolecules. Okay, so biomolecule is compounds made up of carbon. A hydrocarbon is made up of carbon and hydrogen. And when you talk about biomacromolecules, they are very big biomolecules. So as the definition goes, organic compounds such as proteins, nucleic acids, uh, polysaccharides, lipids. So these are acid insoluble, and with the exception of lipids, others like proteins, nucleic acid, and polysaccharides. So they have a molecular weight in the range of 10,000 daltons. Okay. So protein, nucleic acid, and polysaccharides, they have a molecular weight of 10,000 daltons. So that's why they're called biomacromolecules. They're huge. So when you look at all the biomolecules which are present in the living organism, so you have two types. One, those which have a molecular weight less than 1,000 dalton. Okay, so normally when you look at biomolecules, they're expressed in daltons. We don't use kgs or we don't use pounds. Okay, so two classes of biomolecules, ones which have a molecular weight less than 1,000 dalton, and so these are called the micromolecules. And then or simply we can call them biomolecules. So we can either call them micromolecules or simply biomolecules. And the other category are called the macromolecules or the biomacromolecules, and they have a molecular weight which is above 1,000 dalton. Okay, all right. So now lipids, if you take the example of lipids, lipids are basically coming under they're not strictly macromolecules. So lipids don't have such a high molecular weight. Okay, so here, so this table is showing the average composition of a cell, and you can see that water is 70 to 90%, proteins occupy 10 to 15%, carbohydrates 3%, lipids 2%, nucleic acids 5 to 7%, ions are occupying 1%. So now we come to proteins. As I said, proteins, they occupy 10 to 15% of the cellular mass. Okay. So proteins are polypeptides. They are polypeptides. So they are basically linear chain of amino acids linked by peptide bonds. So first we'll go to this basic concept that proteins are made up of amino acids. And we have already, in our previous lectures, we have already studied what are amino acids. So just a brief recall of what are amino acids. So basically, amino acids are, this is what an amino acid looks like. The amino acid has a central alpha carbon. This is the central alpha carbon, and this central alpha carbon is joined by different kind of functional groups. We have the carboxyl group, that is the COOH group, which gives the acidic property. And then we have an amino group, which is the NH2 group. There is a hydrogen here, and there is a R, and this R varies from amino acids to amino acids. So there are 20 different kind of amino acids, and this R keeps on varying. So this is basically the picture of the 20 different kind of amino acids, which can be again categorized into non-polar, then positively charged amino acids, polar amino acids, negatively charged, all depending upon the side chain. Or the simplest amino acid is glycine, which has H as the side chain, right? It can be as complicated as lysine, arginine, glutamic acid. So these are the 20 different amino acids based on the R group, which is attached to the central carbon, the alpha carbon. Okay. So now, very important thing, that proteins are made up of amino acids. So two amino acids, they join together, they form a peptide bond, and like that, many amino acids, they join and they form a long a chain polypeptide, which is basically the protein. And as I said, there are 20 different kinds of amino acids. So there is different thousands and thousands of different proteins are formed from the different permutation and combination of these 20 amino acids. Now, that's why a protein is called a heteropolymer. So protein is called a heteropolymer. It's not a homopolymer. A protein is made up of different types of amino acids. A protein is not made up of same kind of amino acid. Had a protein been made up of same kind of amino acids, we would have called it a homopolymer, but this is a heteropolymer. Okay, so it is not a homopolymer. Okay, now proteins can be classified, or we can say the amino acids are classified into two categories. We have essential amino acids and we have non-essential amino acids. Okay, so what are essential amino acids? So essential amino acids are acids which are not produced inside our body, hence we need those in our diet. Our diet has to have those amino acids because our body does not make the essential amino acids. Non-essential amino acids are amino acids which our body is making. So since our body is making those, since our body is synthesizing the non-essential amino acids, that means we don't need non-essential amino acids in our diet. Okay, so therefore amino acids can be essential or non-essential. The latter are those which our body can make. Latter means the non-essential amino acids are one which body can make, while we get essential amino acids through our diet, through our food. So here is a table which is showing us the essential and the non-essential amino acids. You can see that these are the essential amino acids. So we'll quickly go down the list of the essential amino acids. We have histidine, we have isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. All are essential because our body can't make it, and we get them from diet. We get them from diet. Non-essential amino acids are those which our body can make. So we have alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, and proline. Again, we can look at the amino acid chart and we can figure out what kind of amino acids are there, whether they are actually non-polar or polar or positively charged or negatively charged. If you look at this Venn diagram, we can see that alanine, asparagine, aspartic acid, and glutamic, they are strictly non-essential. Essential amino acids, we have histidine, we have lysine, methionine, phenylalanine, we have threonine, we have tryptophan, we have isoleucine, leucine, and valine. These are strictly essential. We have a middle category like the arginine, cysteine, glutamine, threonine, tyrosine, glycine, proline, and serine. These are conditionally essential. What is the meaning of conditionally essential? That means these are basically, they are non-essential amino acids. They're basically non-essential means our body can make them, but during any kind of illness, when the body is suffering from some kind of illness, then the body is unable to make these amino acids. And when the body is unable to make these amino acids, they become essential. So they're conditionally essential. In a normal healthy person, these are non-essentials. Body is able to synthesize them. In a person who is ill and is unable to make them, then it switches to the essential category. Okay. Now, what are the functions of the protein? Proteins have various functions in our body. Proteins carry out many functions in the living organism. Some are actually acting as some transport nutrients across the cell membrane. So we call them the transport proteins, uh, right? So these proteins are present in the cell membrane, and they help in different kind of transports. That can be an active transport, that can be a passive, that can be a facilitated diffusion. um, and then some proteins, they fight infectious organisms, and some proteins are hormones. Some proteins, they act as enzymes. In fact, all enzymes are proteins, but not all proteins are enzymes. So this is a very famous sentence which says all enzymes are proteins, right? So all enzymes are made up of proteins, but not all proteins are enzymes. Okay. So proteins, they act as enzymes, and we know what are enzymes. Enzymes are the biocatalysts. They enhance the speed of a reaction. They help and a reaction to be carried out at a lower temperature, right? So they basically lower the activation energy. Okay. Then we have collagen. It is the most abundant protein in the animal world. Collagen, under the skin, we have the collagen, right? So which makes the skin very tight, right? So these are the collagen fibers. They are basically also called the white fibers. They are also called the inelastic fibers. And if you if you read the tissue chapter, there we know that the connective tissues, they have collagen in their matrix, and these are the white inelastic fibers which are made up of proteins. Okay, and they are abundant in the animal world. Another protein name is ribulose bisphosphate carboxylase oxygenase. Ribulose bisphosphate carboxylase oxygenase, in short form called rubisco. This rubisco is the most abundant protein in the whole of the biosphere. Where do we find rubisco? We find rubisco in the chloroplast, right? So basically, quickly we'll just so in the chloroplast of the, so when you look at the plant, this is a leaf. Inside the leaf, we have the mesophyll cells, right? Inside the leaf, we have mesophyll cells. Inside, so this is called the mesophyll cell. Inside the mesophyll cell, we have the chloroplast. Inside the chloroplast, we have these kind of structures called the grana or granum. Each of these are called the thylakoids. Thylakoids are joined by the stroma lamella, and then we have the background. We have the stroma, and this is the background stroma. And in the stroma, we have the reaction that is called the Kelvin cycle is going on. Okay, so there is a Kelvin cycle going on. Kelvin cycle, CC, is going on. And in Kelvin cycle, what is happening? Very briefly, this is not the topic of this. So carbon dioxide is assimilated by ribulose ribulose bisphosphate, and this reaction is catalyzed by the rubisco enzyme. This rubisco enzyme is present in the stroma of the chloroplast, and where RUBP is accepting carbon dioxide, there is formation of PGA, phosphoglyceric acid. PGA is then reduced by ATP and NADPH to form the GA3P, glyceraldehyde triphosphate, right? And out of this glyceraldehyde triphosphate, glucose is formed, and rest of the glyceraldehyde triphosphate is recycled or regenerated back to RUBP. So this is the Kelvin cycle. But the important point, so this is basically part of the dark reaction, right? Photosynthesis has two kinds of reactions. Photosynthesis has two reactions: the light reaction the light reaction and the dark reaction. And it is in these reactions that we see glucose is made, right? Oxygen is liberated, right? This is a whole photosynthetic reaction. And so Kelvin cycle comes under the dark reaction. And in this Kelvin cycle, we have Rubisco is the enzyme that carries out this initial step of carbon fixation. And this Rubisco is basically an enzyme, means it's a protein. It's the most abundant protein on the face of Earth, right? In animals, it is the we have collagen. In case of plants, it is Rubisco, which is the most abundant protein in the whole of the biosphere. What is biosphere? Biosphere comprises of atmosphere, hydrosphere, and we have lithosphere, and all the living organisms present in these three sectors of the planet, right? So all the living organisms living in the atmosphere, lithosphere, and in the hydrosphere. Okay, so in biosphere, Rubisco is the most abundant enzyme. Okay, or protein. So now, next we move to. So here we have different kinds of proteins and their functions. The function of collagen is intercellular ground substance. Function of trypsin is acting as an enzyme. We have insulin functions as a hormone. Antibodies are types of protein. Their function is to fight infections. Receptor proteins, they accept some kind of ligand. So receptor proteins are found in our eyes, are found in our nose, in our tongue. So they have sensory reception like smell, taste, hormone. GLUT4 is basically a type of it's a kind of protein which enables glucose transport into a cell. So it helps the insulin in actually transferring the glucose from the blood into inside the cell. Okay, so these are different kinds of proteins and they have different kind of functions. Very quickly, we'll look at the different role of protein in our body. And what do we see here is first, the most important role of the protein is acting as an enzyme. So proteins, they act as enzymes, and that means they are acting as the chemical catalyst. They're speeding up a certain biochemical reaction. Without the presence of enzymes, a biochemical reaction will take a long time. And not only that, our body has a temperature of 37 degrees Celsius. So any kind of biochemical reaction has to be carried out within the 37 degrees Celsius. So this actually is possible because of the presence of these kind of enzymes, which lowers the activation energy. And by lowering the activation energy, they make it possible for any kind of reaction to take place at a lower temperature, which is the temperature of our human body, which is 36 or 37 degrees Celsius. Okay, or in Fahrenheit, it's 98 Fahrenheit. Okay, so they are acting as enzymes and the chemical catalysts that speed up and regulate virtually all chemical reactions in the cell. And we human beings are basically biochemical machines, right? There are thousands of biochemical reactions happening inside a cell, right? And we cannot survive without biochemical reactions. Proteins also function as structural proteins, which is found in the hair. We have the keratin, structural proteins, okay? And fibers that make up the connective tissues, like the tendons. What are tendons? Ligaments. Tendons are something which are connecting the muscles to the bone. So tendons, they connect muscle to bone. And then we have ligament. Ligament is very elastic. Tendon is non-elastic. Ligament is elastic. It helps to connect bone to bone, right? Between two bone, there is a ligament, right? It's connecting bone to bone. This is actually also a structural protein, right? Then we have muscle. Muscle cells. When you look at a muscle cell, they're packed with contractile proteins. What are these contractile proteins? In a muscle cell, contractile proteins are, there are two kinds of contractile protein. We have actin, which is a thin protein, and then we have myosin. So if we look at a skeletal muscle cell, which is basically a columnar cell, elongated cell. There is lots of myofibrils, and these myofibrils, inside the myofibrils, there are these kind of proteins, contractile proteins, actin and myosin, which slide on each other. Because of this sliding, we see the muscle contraction happening. Okay. Also, we have proteins which include defensive proteins such as antibodies. Antibodies, they defend us. If any kind of infection comes inside our body, antibodies are preventing us, protecting us. So such as antibodies of the immune system. And then we have the signal proteins, such as many of the hormones, right? Hormones, a lot of protein-based hormones, which help to control and coordinate between different kind of organs, right? Hormones help in communication between different kind of organs. Okay, hormones, they are carrying through the blood, and then they go to the target organs, and they execute different kind of functions, right? So these hormones are helping in control and coordination in the human body. And other chemical messengers that help coordinate body, they're also made up of proteins. So that is the function of proteins. Next, we go to other roles of proteins are acting as receptor proteins. When I say they are receptor proteins, that means they are found on the cell membrane, and they are found on the cell membrane, and they accept some kind of ligands from outside, and then they carry out a set of reactions. So receptor proteins build in the cell membrane, transmit signal into cell. That is another function of protein. Hemoglobin is it's a protein. It's a mega protein. It is made up of 4 different types of polypeptides, and the function of hemoglobin is they transport oxygen. They bind to oxygen, and they carry it to the tissues, where the tissues are getting oxygen. Where is hemoglobin present? Inside the RBC, the red blood corpuscles. Inside the RBC, we have this hemoglobin, and it's a big protein. It is made up of it's a protein. It's made up of it's a protein complex, you can say, because it is made up of 4 different kinds of polypeptides. Alpha 1, alpha 2, beta 1, beta 2, 4 different kinds of polypeptides. Okay, and each polypeptide can accept an oxygen atom, and it can transmit that to the tissues of our body. That's how, when we breathe in the oxygen, that oxygen is carried by hemoglobin, and they transfer that to the different kind of tissues in our body. And also, we have protein hormone like insulin. Insulin helps in transferring the glucose that is present in the blood into the cell. So when we have a sugary diet, and there is high level of glucose in the blood, which we call hyperglycemic condition, the insulin is secreted from the pancreas, and this insulin is a protein hormone. They help in transporting or transferring the glucose that is present in the blood vessels into the body cells. In this way, we don't have a hyperglycemic situation. Okay, diabetic people whose insulin is not properly working, they have this issue. They have high amount of blood glucose level, and this is called hyperglycemia, that leads to a lot of organ damage. Also, we have storage proteins like the ovalbumin. Ovalbumin is basically the egg white. You have the egg, middle yolk, outside there is a white. So the protein of the egg white, that's called ovalbumin, and which serves as a source of amino acids for the developing embryo. So this egg white is a source of amino acid for the developing embryo. So it is storing these amino acids. Okay. Also, milk proteins provide amino acids for the baby mammals. Like the baby mammals, they're drinking mother's milk. So milk has a lot of protein, and these proteins are actually provide amino acids for the baby mammals. Okay. Plant seeds. If you look at a plant seed, it contains storage proteins that nourish the developing embryo. Okay. So in the endosperm of the plant seed, inside the plant seed, there is the endosperm, which has these kind of protein, and which is supplying to the developing embryo. Okay, so all these are different kind of functions of protein. Now, one thing we have to understand is the structure of protein, and proteins have 4 different kinds of structure. Okay, so when you look at a protein, it has 4 different kinds of structures. We have We have a primary structure, we have a secondary structure, we have a tertiary structure, and a quaternary structure. So we need to understand these 4 different structures of protein. Now, so proteins, as I said, are heteropolymers, right? So when I say they are heteropolymers, this means it is made up of different combinations of amino acid. It is not made up of one type of amino acid. Okay, so different combinations of amino acids are there. Okay, and so these amino acids are joined one to another by the help of a peptide bond, and there is a string of amino acids. So basically, so what we can do is we can clearly go and have an idea of how this reaction takes place. So let's say we have, this is one amino acid. This is R1. This is the C. This is the COH group. This is the NH2 group. And this amino acid is now binding with the second amino acid. And this is the hydrogen. This is the R2. And this is the COOH. Quickly, let me draw the hydrogen here. And then we have the third amino acid, NH2CHR3, and here we have COOH. Now, by dehydration reaction, that means this water molecule and this water molecule, they escape, and a peptide bond is formed between amino acid 1, 2, and 3. So how does it look like? Basically, this is how it looks like. The CO is now bonded. I'll give a black line here. It's bonded with the NH. So this CONH is a peptide bond, and then CCO. Again, I'll give a black line here. So this is basically the peptide bond formation between these 2 amino acids. And we can draw the rest of the amino acid or the protein like that. And this is basically obviously the R1 group. This is the H group. And this is basically the peptide bond formed here. This is the peptide bond formed here. And you can see how so here, basically the alpha carbon. So there is a H, there is a R3, there is a COOH. Again, this one can form another peptide bond with another actually amino acid. Okay. So here we have, let me just complete this whole thing. So this is basically R2 here. So this forms, so these are the peptide bonds, that the peptide bond. Okay, this is also a peptide bond, peptide PB, peptide bond. And this can keep on adding and keep on adding. So basically I can draw a third bracket and put an N, and N can stand for 1,000 to 10,000, even higher number of amino acids. And this is basically called the primary structure of protein. So when I say primary structure of protein, so basically this is the amino acids joined to one another to form a long chain polymer, which is called the polypeptide, because each of these are peptide bonds. So this is called polypeptide, and they can be almost 1,000 polypeptides, 10,000 polypeptides, forming a primary structure of a protein. Okay, so when we look at the primary structure of a protein, so let's see some diagrams of how the primary structure of the protein looks like. So here we can see the primary structure is made up of, let me just zoom it. So you can see the primary structure is formed of N and CO. This is the peptide bond, again N and the carboxyl group. So I'll keep on pointing here. I'll keep on pointing the primary structure. So here we have the peptide bond between the N and the carboxyl group, right? Again, between the N and carboxyl group, and this is the primary structure of the protein, which is basically showing the peptide bonds, right? Now, after the protein has 4 different kind of structures. After the primary structure, it has something called the secondary structure. So what is a secondary structure? Secondary structures can be 2 types. Okay, so let's try to draw the secondary structure. So these are, after the primary structures are formed, which is basically 2 amino acids joined together forming a peptide bond. These can now take different kind of folds. And when I say folds, the folds can be, so the secondary structure can be of 2 types. We have the alpha, helix, the alpha sheet, the alpha helix, and we have the beta pleated sheet, or we can see the beta sheet. So there are 2 kinds of secondary structures. What are secondary structures? So going back to, so the secondary structures are, so folded, the secondary structures are, when the primary structure is folded in the form of a helix. You see, when the primary structure is folded in the form of a helix, right? Similar to a revolving staircase. The primary structure is folded in a revolving structure, right? Of staircase, revolving staircase. That's what we call the helix. Okay, and some portion of the protein thread are arranged in the form of helix. So some portion, not all the protein, but some portion is arranged in helical form, right? Right-handed helix. So how do we show that? So let's say this is the protein. This is the, let's say, the primary structure of a protein, which is so I say the primary structure is amino acids and peptide bonds. So we have amino acid, amino acid, amino acid. So 1, amino acid 2, amino acid 3, and then suddenly we have a helical structure in the middle. Again, from here we have amino acid, amino acid, amino acid, right? So this part is called alpha helix secondary structure. Alpha helix secondary structure. This part is called the alpha helix secondary structure. And then again, after some time, you will have these kind of, okay. So these are called the beta. We draw the beta pleated sheet in this arrow form. Okay, so these are called the beta sheet. Again, they are type of secondary structures in the protein. And then again, after that, again from here, amino acid, amino acid. Okay, so we have primary structure, which is basically just the amino acids joined by the peptide bond. So we have the primary structure. We have a secondary structure here in the form of alpha helix. This is the secondary structure. Again, then we have in the middle, again, a primary structure PS, and then again, we have a secondary structure, the beta sheet. So this is again a secondary structure, beta sheet. Again and again, then we have a primary structure. So that's how the protein is. This is the second level shape of the protein. So we can see another picture, which will give an idea. Here. So we can see how this is the primary structure of the protein, and then let me use a pointer. So this is the primary structure of the protein, and then suddenly at this part we have the alpha helix, which is the secondary structure of the protein, and then again we have a small primary structure here, and then again we have the beta pleated sheet. You can see how it looks like an arrow like that. So now when we go deep into these structures, you see how the alpha helix is like a helix, right? How it is like a helix like that, right? And now observe carefully. This is basically the primary structure. You see how the primary structure is there. This is the carboxyl group. This is the alpha carbon. This is the NH group, right? And again in the next, so this is one helix, and it's going back like that, another helix, and then going back another helix, right? That's how it is. We can draw that. So basically, one helix, and then the back side, and then another helix, and then there's a back turning, and then another helix, right? So like that, it is a helix. So here, with a different color, I'll draw. Here we have the CHCOO right? We have the NH, and you can see this NH here. It is hydrogen bonding with the lower. So here there is a lower carboxyl group, and between these two, in orange, there is a hydrogen bonding happening, right? There is a hydrogen bonding. So this is basically the NH, and this is the CHR. This is the alpha carbon, right? This is the alpha carbon. This is the NH. This is the CO. This is the peptide bond, right? Here is the peptide bond. And you see how between these two, there is a hydrogen bond present, right? So now you can understand that this is a hydrogen bond between the H and the O, because H is electropositive and O is electronegative. So between them, there is a hydrogen bond between that, right? Again, we see there is a hydrogen bond here, and these hydrogen bonds are making this alpha helix very strong structure. It is not breaking down, right? Because of this hydrogen bond, okay? Also, if you look at the beta pleated sheet, there also we see hydrogen bond. You see this is the primary structure. You can see, and how you see how like a beta pleated sheet, right? Like this, okay? So this is how the beta pleated sheet looks like, right? So zigzag, right? If you look at zigzag, right? This is zigzag kind of shape. That is the beta pleated sheet. That's a kind of secondary structure, okay? And again, we have the primary structure here. You can clearly see the primary structure, which are the amino acids joined by the peptide bond. And you can see how the NH of this above sheet and the CO carboxyl group, they are in a hydrogen bond. Again, the carboxyl group and the NH in the two parallel beta pleated sheet, how they're in the forming hydrogen bond. Again, there is a hydrogen bond formed here. There is a hydrogen bond formed here. So many hydrogen bonds are formed between two parallel beta pleated sheet, and this makes the beta pleated sheet very. So these hydrogen bond, they make this very strong. Okay, very strong. And how much strong I'm telling you? Let's say the spiders, they make spider web, and that is basically a protein having lots of beta pleated sheets. So the spiders, they are from the saliva, they're making a protein. With the help of this protein, they are making the spider web. This spider web has, if you look at the, if you clearly, if you observe them, we go deep into the protein structure level, and if you observe them, then the spider web has lots of beta pleated sheet, and that makes the spider web very strong. If you take an equivalent spider web and an equivalent steel wire, if you take the equivalent weight, then we will see the spider web is more stronger than the steel wire. It sounds funny, it sounds something which is hard to believe, but that is actually the truth. It is because the spider web has these beta pleated sheet and they're bound by the hydrogen bond. It makes the spider web so strong, stronger than a equivalent mass steel wire. Okay. So now, so we have the primary structure, we have the secondary structure, which is the alpha helix and the beta plated sheet, and then we also have the tertiary structure. Okay, so now let's see what is a tertiary structure. So now this is a picture, very nice picture, which shows the primary structure, which is basically the amino acids joined by peptide bond. And then you see how some part of them can form the alpha helix, right? This is the alpha helix, and we can also see the hydrogen bonds present. Some part of the proteins, some part of the primary structure can form these beta pleated sheets, parallel sheets, right? One beta pleated sheet, another beta pleated sheet, now parallel there, and then there's how the hydrogen bonding happening, which is making this beta pleated sheet so strong. Now this now further forms a three-dimensional shape, right? So now you can imagine first a ball and a string, which is the primary structure, and within the primary structures we have these kind of hydrogen bonded areas called the alpha helix and the beta pleated sheet, right? And then we have these further have a three-dimensional shape, which is called the tertiary structure, and this tertiary structure is very, very important. If proteins are unable to form tertiary structure, then the protein cannot be functional. In order for a protein to have a proper function, just simply having a primary structure and a secondary structure is not enough. That protein should also have a three-dimensional structure, which is called the 3D structure. And depending upon that particular 3D structure, a protein can bind, can catalyze a particular reaction. They can bind to a particular reactant. So here, let's say a protein is looking like this is the protein, a huge protein, and so it has a particular active site, right? This is called the active, or maybe we can draw in a different, nicer way. So this is protein, and let's say this is the active site of the protein. So this is a 3D structure of the protein. You can see the 3D structure of the protein, and it can only bind to a reactant which fits this active site. Okay, so this is the active site of the protein, and this here, this is the reactant, and the reactant is binding to the protein because this protein has a active site which has the same shape as the reactant. Okay, that's why proteins are, or enzymes are, specific for their reactants, right? So this 3D shape is very important. If this protein hadn't had this 3D shape, then this reactant wouldn't have been fitting inside the active site and the reaction wouldn't have been carried out. Okay, so the protein function, for the protein to be functional, it should have a tertiary structures. Now sometimes we have these complex proteins, huge proteins, multi-protein complex. One example is hemoglobin. Their different tertiary structures, they come together and form a quaternary structure. So many tertiary structures, they come together and form a quaternary structure. This happens in some, it doesn't happen in every protein. Some proteins are multi-complex proteins where they have tertiary structures coming and forming the quaternary structure. So here, if you see at this diagram, so this tertiary structure and another tertiary structure, they're coming together and they're forming the quaternary structure. The best example of this is, so here we can see this picture, how we have the primary structure, then we have the secondary structure, alpha helix, and the beta pleated sheet. Then we have the tertiary structure, which is the folded 3D structure required for function. And sometimes in the multi-complex proteins, different tertiary structures come together and form the quaternary structure. So if we look at the example is hemoglobin. Look, this is actually hemoglobin has 2 alpha chains and 2 beta chains. Each of this alpha chain is basically a tertiary structure, right? So this is one tertiary structure, this is one tertiary structure, these blue ones are tertiary structures, how they come together and they form a quaternary structure, right? All right, I think that's for today. just a small recap of what we did. We learned several things about what are proteins, and proteins are basically biomacromolecules because they have size over 1,000 daltons. Okay, they have weight almost like 10,000 daltons. Very, very big molecules. Okay, so proteins, nucleic acids, polysaccharides, they're all called biomacromolecules. Okay, lipids are not having so much high molecular weight, unlike proteins, nucleic acids. And then we learned about proteins are made up of amino acids formed by peptide bond. There are 20 different kinds of amino acids. So we learned about that, and this is the amino acids. Okay. We learned about what are essential amino acids, non-essential amino acids, also conditionally essential amino acids. We learned that. And then we learned about the function of protein, which ranges from various functions, functioning as enzymes to transport nutrients across the cell membrane to fighting infections. And we learned about the most abundant animal protein, which is collagen, and the most abundant protein in the biosphere, which is the rubisco. And we learned about proteins, their functions. And then we learned about the protein structure, which is the primary structure of a protein, which is a secondary structure. One thing I forgot to mention today is the N-terminal end and the carboxy terminal end. So when you look at a protein primary structure, the left side of the protein, the left side has the amino group, right? If you look at a very long primary structure, you will see the left side has the amino group and the right side has a carboxy COH group. So this green patch here, this is the N terminus of a protein, and this pink box here, it shows the carboxy terminus. So if you just kind of join two amino acids and if you keep on joining them, at the end of this long polypeptide, this long primary structure, on the left side, you will see the N terminus, and in the end, you will see the carboxy terminus. Very, very important to understand this concept. And also, we learned about how proteins form the secondary structures, which are the alpha helix and the beta pleated sheet, how there is hydrogen bonding, how the hydrogen bonding makes these structures so strong. And then we know how these form a 3D shape of the tertiary structures, and how this third tertiary structure is important for maintaining the function of the protein. And finally, we learned about how these tertiary structures come together and they form the quaternary structure. Example, we learned in the case of hemoglobin. Here, how hemoglobin, different kind of tertiary structures, they come together and form the quaternary structure. Hemoglobin helps in transport of oxygen. So we'll stop here and we'll meet for other classes. Bye-bye.